The overall goal of this procedure is to recover individual or agglomerated cells, termed putative bioparticles, from forensic touch DNA evidence and obtain a DNA profile of the donor. During the commission of a violent crime, a small number of cells are transferred from an individual to another individual, such as when an assailant grabs a victim's wrist. In order to recover the small number of cells, first use a low-adhesion gel film attached to a microscope slide to sample the contact area. The second step is to stain the transferred bioparticles for easier viewing under the microscope. Next, isolate the bioparticles from the gel film using micromanipulation. Collect the water-soluble adhesive at the tip of a tungsten needle and use it to remove bioparticles of interest from the gel film surface. The final step is to carefully transfer the individually collected bioparticles to a PCR amplification mix at the bottom of a PCR collection tube. Ultimately, enhanced DNA typing methods are used to obtain a DNA profile of the donor of trace DNA evidence.
In my lab, we focus on the genetic analysis of the small numbers of cells that are transferred during the commission of a violent crime against a person between one individual and another individual, or between an individual and an object. Traditional methods often fail to recover the DNA profile from such small numbers of cells. Here we have developed a simplified method for the physical recovery and genetic analysis of such small numbers of cells, either individual cells or agglomerates of cells that can be incorporated into any forensic